I'd like to thank the organizer and the RGC for inviting us and give us this opportunity to share some of our research with you. So in 1997, my group first reported that during pregnancy, a fetus would release its DNA in the bloodstream of the mother. And this DNA is present very early on from the first trimester. And actually by the 10 weeks of pregnancy, it reaches some 15%. And then after delivery, then the baby fetal DNA is removed very quickly. So we have explored the prenatal diagnostic application of these approaches. And one important disorder that many pregnant women undergoing prenatal testing would be concerned with is the fact that the baby might be affected by chromosome disorder like Down syndrome. So in 2008, our group reported a method whereby by randomly sequencing millions of DNA molecules in plasma and to map each of those molecules back to chromosome from which it come from, we can work out a proportional representation of different chromosomes in plasma. And by using this method, we find that the diagnostic accuracy of it for Down syndrome is over 99%. And because of its accuracy, this test was introduced in the clinical practice in 2011. And to date, over 10 million tests are done every year. So for this TRS project, we're very interested in some of the mechanism and the biology of this phenomenon. And one of that is that the circulating DNA are basically short fragments of DNA. So we have been very interested about the mechanism whereby this fragmentation takes place. We think it's likely that this fragmentation is taking place because of some enzymes, such as nuclease. So if that's the case, then one way we can tackle this is to basically knock out nuclease genes in mouse models. Now, for example, if you take some blood sample from a mouse, a wild-type mouse, and then you do Illumina sequencing. So you take the DNA and you add on the Illumina sequencing adapter and you run it on a gel, you see a predominant band around 300 base pairs. So this is consisting of 140 base pair of the adapter plus the 160 base pair, which is the plasma DNA. And this 160 base pair basically is a mononucleosomal DNA. Now, so we're thinking that if we knock out nuclease, and then if we look at literature, many people think that in the blood, DNase 1 is a major nuclease in our blood. So the first thing we ask is, if we knock out DNase 1 gene in mice, would we change the size of plasma DNA? And strangely, when we knock it out, we actually find that the size of DNA molecules are basically remain unchanged. And so we wonder, are there any other nuclease which we can do that? And one nuclease we've been thinking about is a nuclease called DNase 1-like-3. And some people also regard this as DNase gamma. So basically, there's a nuclease which digests chromatin. And because circulating DNA is basically circulating chromatin, so we knock this gene out, and lo and behold, we start to see longer DNA molecules. So you can see those which are dinucleosomal, some which are 3 nucleosomes, and some are 4 nucleosomes. And then if you do the double knockout DNase 1-like-3 plus DNase 1, then the pattern doesn't change. So we published this in 2019. And so this is a graph which will plot basically the size distribution of the DNA concentration in plasma in mice in which the DNase 1-L3 gene has been knocked out versus the wild type. So you can see that the knockout model have much longer DNA molecules. So after we have established that DNase 1-L3 like to cut this plasma DNA, so the next question we ask is, are there any sequence that it particularly like to cut? So we actually look at the N nucleotide motif of the plasma DNA. And to start off with, we look at 4 nucleotides and then try to tally the concentration of those 4 nucleotide motifs. Because each position of 4 nucleotide motifs, there are 4 possibilities. So 4 to the power 4 is 256. So in other words, if the motif frequency are random, then basically each motif will be present at a concentration of 1/256, which is 0.39%. Now, interestingly, when we do the experiment, we find that in wild-type mice, the motif frequency of the top motifs is actually much higher than 0.39%. It's actually 1.5%. And furthermore, when you knock out the DNase 1-like-3 gene, then those motif frequency actually change significantly. And also, if you're observant, you'll see that actually the top 6 motifs here all start with CC. So basically, our working hypothesis is that the DNase 1-like-3 enzyme like to cut DNA molecule with CC. So now when we pool those 6 motifs with CC together, we find that those plasma DNA molecules represent 7.5% of all the circulating DNA population. Now, if you knock out the DNase 1 gene, the motif frequency doesn't change. But on the other hand, if you knock out DNase 1-like-3 gene, then the motif frequency basically is halved. When you do double knockout, it's still halved. So we wonder what would happen in pregnancy. So we look at a pregnant mouse model in which we have female black 6 mouse, in which we have knocked out both copies of DNase 1-like-3 gene. And we cross this mice with male BALB/c mouse, which is wild type. So the fetus will be heterozygous, so have 1 copy of DNase 1-like-3 gene and 1 copy knockout. So assuming that the cellular trafficking of cell-free DNA is the same in mice as in human, then the fetus should release the fetal DNA into maternal blood. Now, just to remind ourselves what happened in non-pregnant situation. So basically, those 6 motifs together represent 7.5% of all circulating DNA. You knock out the DNase 1-like-3 gene, the motif frequency is halved. If you go to pregnant situation when the mother and baby are the same, so in other words, wild type, and knockout, then the motif frequency is the same as non-pregnant situation. So now come the interesting bit. When you have a pregnant situation, when the mother is homozygous knockout and the fetus is heterozygous. So you can see that in this situation, the fetus is able to partially rescue the phenotype of the mother. Now, because the fetal DNA has this paternal BALB/c background, you can just specifically look at the fetal DNA mother's blood. You can see that the motif frequency of those fetal molecules is even more normal. So basically, our conclusion is like that. So in this pregnancy model, the baby mouse will release 2 things in mother's blood. The first, because it has 1 copy of DNase 1-like-3 gene, is able to manufacture the enzyme. And so they will release the enzyme into maternal blood. And this enzyme is going to cut the mother's DNA, so resulting in this partial normalization of the motif frequency. And the second thing is that because the enzyme is present or originate inside the fetus, so it will also do some local cutting of fetal DNA. So that's why the motif frequency of the fetal DNA is more normal than that of the mother. Now, so we think that this work is important because it's a first link between nuclease biology and circulating nucleic acid. And also we show that this phenomenon will have implication in physiologic conditions like pregnancy, and also in pathological condition when you have a lack of this enzyme gene. But of course, the data is generated from mouse model. The question is whether you also apply to human beings. So in that regard, we have established a collaboration with Stefano Voppi from Italy and Linda Hiraki from Toronto. And then we're able to actually find a number of individuals who are homozygous for mutation of a DNase 1-like-3 gene. And actually, those individuals will develop a systemic lupus-like disease. Now, so when we profile the size of those of this project, we actually find that in control individuals, the highest peak size of the circulating DNA is 160 base pair, which is the size of a nucleosome. When we look at the parents of the patient, so parents are heterozygous carrier of the mutation, so they don't have disease themselves. So the size profile of those individuals are similar to those individuals who are completely normal. But however, when you look at the individual with homozygous mutation, they can see that the pattern, basically, they show an increased amount of longer DNA, which is very similar to what we've seen in the mice. And furthermore, when you look at the motif frequency, for example, you look at the CC motif here, and you look at the healthy control of people with systemic lupus but without the mutation, or people with juvenile idiopathic arthritis, or people with transplantation, then see basically they all have this predominance of the CC motif, just like what we see in the wild type mice. But on the other hand, on the right-hand side here, in individual with homozygous mutation of the DNase 1-like-3 mutation, they can see that basically there's a reduction of the CC motif frequencies in the plasma DNA, which once again, just like the knockout mice. Now, when you plot those 6 CC motifs together in the control subject in human beings, the concentration is around 10%. But in the individual with homozygous mutation, there's about half. So once again, it is very similar to what we see in the mice. So basically, it gives us confidence that what we found in the mouse model will have implication in human subjects. But of course, the question is, what sort of application are we talking about? So we think actually that this motif frequency is a new type of diagnostic marker that we can use. Now, for example, in the pregnant plasma, there's basically a mixture of fetal and maternal DNA. So in many of the prenatal diagnoses that we're doing, this existence of this maternal background will complicate matter for us to do prenatal testing. So for many years, people have been trying to see if there's any way we can differentiate the fetal and maternal DNA. And now, with motif frequencies, we think that we have a way. For example, on the right-hand side here, basically, you're trying to differentiate the fetal DNA, which is plotted here in red, and the shared DNA, which is fetus and mother, so but mainly from the mother. So you can clearly see, basically, by using motif frequency, we can clearly separate the fetal DNA and the mother's DNA in the maternal plasma. So we think actually this will have a lot of interesting prenatal testing application, for example, in single gene disorders, etc. And this work has been published a few months ago in Cancer Discovery. And incidentally, we also find that this motif frequency can also be used as a generic cancer marker. You can also allow you to tell the DNA released from a transplanted organs, because basically each tissue type within a body will have their own motif frequency. So, but however, we think that this DNase 1-like-3 is probably the tip of the iceberg. There are probably a whole group of nucleases which are doing a similar job. Now, and we think now with an improved understanding of the biology and the technology to assay for this change, we think it might be time for us to take another look at DNase 1, which previously, just by looking at its size, we don't see a difference. But we think now, by using motif frequency and other modalities, we may be able to attribute a role to DNase 1. And we also decide to take a look yet another nuclease called caspase activate DNase. And some people also call this DNA fragmentation factor beta, DFFB, which is involved in the apoptosis. So basically, the study here is a little bit similar to before. So we all look at wild type mice. We also look at mice in which a DFFB gene has been knocked out. Those with the DNase 1-like-3 gene has been knocked out, or DNase 1 gene has been knocked out. But in addition to looking at the fresh plasma, we also take blood from those mice and deliberately leave the blood in room temperature for 6 hours. And we know from previous work that if you do that, then actually the blood cells will release the DNA into plasma. So it let us take a look to see what the freshly released plasma DNA will like. Now, at this time, because we're working on three different enzymes, so the experiment will be much more complicated. So instead of looking at 256 motifs, we decide to simplify it just to look at a single nucleotide motif. So either a plasma DNA ending in C, T, G, or A, just separate into 4 different groups. Now, for example, if you look at wild type mice, you took them blood and freshly and analyzed them. And you can see that the plasma DNA with different A, C, T, and G and N's will have different proportion. So now if you leave the blood in room temperature for 6 hours, then you actually start to see there's some newly released DNA being assayed. And actually those newly released DNA is predominant of A and G variety. But now when we do the experiment again, and this time we actually knock out the DFFB gene, then you can see that actually the change at 6 hours has disappeared. So now as one conclusion here, we can conclude that the DFFB enzyme would like to produce DNA ending with A and G. So in other words, the DFFB enzyme will prefer A and G. And you do the same thing again by using DNase 1 like 3 knockout, and you find that it prefers C. And you do the same thing with DNase 1, and you find it prefers T. And then, of course, you can also repeat experiment by knocking out other nuclease genes, and you can put this together. And actually, this work has basically been recently published in the American Journal of Human Genetics, basically in which we talk about this stepwise model. And actually, the American Journal of Human Genetics have actually recently regarded this paper as one of the best for this year, so which we're very encouraged and pleased. Now, up to now, we've talked about this circulating DNA, and many people regard circulating DNA as being double-stranded DNA. But one question we've been asking recently is this double-stranded DNA is applied to every molecule, or is it possible that some molecule could be partially single-stranded? So we call this a jacket end. But of course, if it is jacket end, then we have to develop some technology to allow us to assay for it. So we think, well, if I repair the jacket ends, and during the repairing step, maybe I can incorporate some marker onto it, which can be detected later on. Now, for example, like unmethylated C. Now, the reason why unmethylated C is a type of marker which is useful is because if you look at CG in the genome overall, 70% of C is methylated. So in other words, if I have a jacket end, if I incorporate unmethylated C, then you should see a reduction in the methylation density. And then after that, I can read out by using bisulfite sequencing. So we call this technology JAK-seq. And interestingly, when we use JAK-seq in plasma DNA, we actually see that for the first time, it's a lot of jacket ends. But in comparison, if you extract the genomic DNA from cells and you sonicate it, then actually the amount of jacket ends is very little. And in addition to just saying whether jacket end is present or absent, we can also use JAK-seq to tell us how long this jacket end is, starting with 0. So in other words, this is blunt end. We find that in plasma DNA, about 11% of plasma DNA are blunt end molecules. But however, there's also plasma DNA molecules with jacket ends ranging from 1 nucleotide in length all the way to over 70 nucleotide in length. And also interestingly, when we look at fetal DNA in maternal plasma, we find that it has more jacket ends than the mother's DNA. And furthermore, those jacket ends are clustered on molecules which is between 130 to 160 base pair. So they are molecules which is just a little bit shorter than that of a nucleosome. So by taking advantage of this discovery, we can basically use the jacket end index to tell us how much fetal DNA we have in the sample. Because the more fetal DNA there is, the more jacket end we'll see. And this is important because this fetal DNA concentration is the #1 determinant of the accuracy of non-invasive prenatal testing. And then we wonder what's the mechanism whereby the jacket end is produced. Now we think it's likely that it is also produced in the same process of plasma DNA fragmentation, which is, as we described just now, is a nuclease-based process. So for example, we look at wild-type mice, and when we knock out DNase 1 gene, then we find that the jacket ends degree has reduced. On the other hand, if you knock out the DNase 1 like 3 gene, then the amount of jacket ends increase. So basically, our working model is like that. So DNase 1 will have a tendency to actually cut one of the strands. In so doing, will produce a jacket ends. But on the other hand, the DNase 1 like 3 enzyme would like to cut both strands, so produce a blunt end. So basically, we think that jacket ends is an intrinsic property of plasma DNA. And then we find that the fetal DNA have more jacket ends than the background maternal plasma DNA. And it has a potential application in non-invasive prenatal testing. And we have also identified the role of nuclease, especially DNase 1, in the production of jacket ends. And this work has just recently been published in Genome Research. So up to now, we've been talking about the fragmentation of plasma DNA. So we now refer to this as fragmentomics. And actually, fragmentomics is a term which originally was first coined to study the fragmentation of peptides. I think nucleic acid fragmentation can be equally well described. So next question, we want to go the other direction. So how intact can circulating DNA be? Now, of course, I won't be naive enough to think that there will be entire chromosome in plasma, but how about a small one, particularly the one from mitochondria? Because a mitochondria has its own genome, and actually this is circular molecules of 16 kilobase. So in previous work, we have shown that mitochondrial DNA present in plasma. For example, you extract the plasma DNA and then put on the Illumina sequencing adapter, you can see some molecules which are a little bit shorter than the nuclear DNA. But however, for this experiment, you basically ligate the sequencing adapter onto DNA molecules. So by definition, all the molecules when assay here are pre-existing as linear molecule. If there's circular molecule there, then you won't be able to attach the sequencing adapter, and so you won't be able to see it. So in other words, we think there's an open question whether there are circular mitochondrial DNA in the plasma. And to assay for those, we need to do some enzymatic tricks. Now, let's say that in the plasma, if there's some linear mitochondrial DNA and some circular one. So the first thing we'll do is to digest away the pre-existing linear DNA by using exonuclease 5. So this will leave the circular DNA intact. And then we need to open the circle, for example, by cutting with a restriction enzyme like MSP1. And then after that, we add on the sequencing adapter and a sequence of molecules. And then we can actually look at molecules in which both ends are flanked by the MSP1 restriction enzyme sites. Just to show you some data. Now, for example, you extract plasma DNA and you don't do any of those enzymatic tricks, then mitochondrial DNA is a very rare occurrence. It's present in about 0.001%. But however, if you cut with MSP1, then you see there's an increase in mitochondrial amount by about 10 times. But on the other hand, if you cut away the pre-existing linear DNA first and then open the circle, then you increase that 10 to the power 4 times. So basically, those molecules are the circular mitochondrial DNA. On the other side, you can also look at it with the DNA molecule flanked on both ends with the restriction enzyme cleavage site. So here is a no treatment, and then this one is treated with the restriction enzymes. You can see there's a massive increase in those DNA. And this one is cutting with exonuclease first and then restriction enzyme. So in other words, both of those are also circular mitochondrial DNA. So another question now, so apart from the presence of both circular and linear mitochondrial DNA in plasma, the next question is, are they coming from different locations? So to answer this question, we actually look at transplantation. So we look at liver transplants and we look at bone marrow transplant. So in this situation, we can basically type the mitochondrial SNPs in both the recipient and donor. So we know that the mitochondria is coming from which individuals. And then we can use the enzymatic trick to tell us whether those DNA are linear or circular. Now, for example, here we're looking at mitochondrial DNA of donor origin. And interestingly, when you look at liver transplant, actually most of the liver-derived DNA are linear. On the other hand, in bone marrow transplant, then both of the donor-derived or bone marrow-derived DNA is circular. So now it's very interesting. So in the transplantation context, the DNA released by liver is linear. On the other hand, in the transplantation context, the bone marrow DNA are circular. And of course, we can debate about where the hematopoietic DNA are originating in. Could they be in circulating platelets, exosomes, or even entire mitochondria? Now, how about pregnancy situation? Because in the literature, nobody has talked about fetal mitochondrial DNA in maternal plasma. And the reason why they didn't talk about that is because it's a very difficult question to tackle. Because a fetal mitochondrial DNA is maternally inherited. So you cannot tell the fetal mitochondrial DNA from the maternal mitochondrial DNA. So to answer that question, we have to have a very special situation of surrogate pregnancy in which the mother is genetically unrelated to the fetus. So in collaboration with a group in Russia, we're able to actually look at that question and show for the first time that indeed there's fetal mitochondrial DNA in maternal plasma. And furthermore, by using those enzymatic treatment, we find that most of the fetal mitochondrial DNA is linear, but the mother side is half linear and half circular. So basically what this shows is it's useful to look at the topology of plasma DNA. And actually now we have more recent work in which we have moved beyond mitochondrial DNA to look at circular DNA of genomic origin, the extracellular circular DNA. And those are also very interesting area. And we find that generally they are longer than the linear DNA. So potentially might have some interesting prenatal diagnostic application. So finally, in conclusion, hope I've convinced you that plasma nucleic acid represent a treasure trove for molecular diagnostics. Now with the effort which is supported by the RGC, we have now already achieved a global adoption of non-invasive prenatal testing. And today we've talked about some new concepts in this area, including fragmentomics. We talk about fragment size and motifs. We also talk about the topology of plasma DNA. We talk about linear and circular molecules. We also talk about this intriguing link to nucleus biology. And finally, I'd like to thank individuals from my group for generating data which I present to you. And a couple of months ago, I was very delighted and humbled to be selected as a winner of this year's Breakthrough Prize in Life Science. So in this regard, I'd like to thank the RGC for the support over the last 2 decades. Without this support, I'll never be able to win this type of international award. So thank you very much for your time.
It is a very comprehensive coverage of the various ways how cell-free nucleic acids can be fragmented. You mentioned about these circular mitochondrial DNA molecules. Do you know if they are actually intact, full-length mitochondrial DNA molecules, or they are fragmented and then became smaller circles, and whether these circles carry out any function, mitochondrial DNA function?
I think from our data at the moment, if we use short read sequencing, we can only guess that. My guess is usually, if I make a scientific hypothesis, I'll probably start with a simpler hypothesis. I think it's simpler to suggest that it is actually the original intact mitochondria rather than a 2-step process. There's some sort of fragmentation and then recircularization. Although I think in the future, if you try to do that with, for example, Pacific Bioscience, those long read sequencing, that will be able to allow us to answer your question conclusively. And of course, there's also a recent paper published by a French group, which actually talk about there are intact circulating entire mitochondrion in the blood. And of course, and those are metabolically active. So if that's the case, I will also guess it's probably the entire genome of the mitochondria is there.
Interesting. So, I mean, other than just studying the phenomenon, maybe in the future, we may study the function of such different fragmented forms of nucleic acid molecules. Talking about the fragments, you mentioned that the end motifs, patterns, may help us to distinguish fetal and maternal DNA molecules. So could you give us some insight in terms of analysis? How might we be able to take advantage of these features and maybe enrich for the fetal DNA signal?
So I think one use is potentially, for example, to look at the SNP composition of fetus. So currently, if you look at SNP, single nucleotide polymorphisms, the most robust is to look at alleles that the baby has inherited from the father, but which is absent in the mother. But in the future, maybe every SNP you look at, you can also add a score based on the motif at the end. And then you can work out algorithm to tell you, okay, the fetus SNP composition across the genome is X or Y. So I think that will be very exciting for single gene disorders.
Thank you. So Professor Lo, we have a question from the audience, Alfred Zhang, asking, do you observe particular pathologies of human subjects with homogeneous mutations? I guess that's referring to the DNA's enzymes homogeneous mutations.
Yeah, that's a good question. The answer is yes. So those individuals develop a type of systemic lupus-like disease, and they have hypocomplement, so complement level is low, and they also have vasculitis. And also, many of them, unfortunately, will proceed to develop renal failure. And that's the reason why we also look at subject with renal transplantation as controls.
And so does that suggest, because DNase 1L3, the absence of it resulted in long DNA molecules, and those individuals have those symptoms, does that suggest that the cell-free nucleic acid molecules might be immunogenic? So what is the chicken and what is the egg?
Yeah, I think that's why this nuclease link to circulating DNA is exciting, because I think it certainly will raise the question of the participation in pathology, the pathogenesis, whether long DNA is bad for the immune system and immunogenic. And of course, it also opened the possibility that you can use nuclease as a type of treatment.